Hi everyone, this is from Vivid Botany. We have here a question where tissues or the cells are described on left side and they are asked to identify which of the following is the correct pair. So we should have here a good understanding of the cells like subsidiary cells, conjunctive tissue, interfascicular cambium, spongy parenchyma. So I will try to explain these cells one after the other. Let us have a look at first subsidiary cells. Subsidiary cells are found in stoma. What is stoma? Stoma is the aerial openings present in aerial living parts of the plant. Aerial living parts of the plant contain openings called as stoma. So in this stoma, I write here, they are having here the guard cells, these are called as the guard cells. Generally, two guard cells are present per one stoma. Right. So these are bean shaped or kidney shaped in dicots and they are dumbbell shaped in monocots. They are having chloroplast also. They are having cellulosic microfibrils that extend radially from one end to the other. These cellulosic microfibrils, they help in stretching the guard cells so that the stoma can open. And they are having chloroplasts, they perform photosynthesis, they have starch grains also. So they are modified epidermal cells that perform photosynthesis and helpful in opening and closing of the stoma. And around this we find another modified cells which are called as subsidiary cells, or we call them as the accessory cells. Subsidiary cells, or we call them as accessory cells. So these are the accessory cells, or they are called as subsidiary cells. So these are the guard cells. Remember, guard cells and subsidiary cells both are modified epidermal cells. Both are modified epidermal cells. But guard cells are generally two in number and subsidiary cells are many in number. That is one difference. And guard cells are having a particular shape, whereas subsidiary cells don't have that particular shape. And this part is called as substomatal cavity. Substomatal cavity, and the central space opening is called as stomatal aperture. Now aperture, guard cells, subsidiary cells, substomatal cavity, together it is called as stomatal apparatus. They are the part of the stoma. So the stoma are meant for gaseous exchange as well as the transpiration. So subsidiary cells are accessory cells, are main part of the accessory cells. Yeah. Now, next, another is conjunctive tissue. What is this conjunctive tissue? We have a line about conjunctive tissue in anatomy, that is in tiers of root made by dicot or the monocot. Let's take the dicot root tiers here. This is the xylem strand that I am drawing here. Generally, we find here 4 xylem strands, 2 to 4 xylem strands that alternate with 4 phloem strands. These are 4 phloem strands. So it is called as tetrarch and exarch. Tetrarch means what? 4 vascular bundles are present. Exarch means protoxylem is towards outside and metaxylem is towards inside. This part is called as protoxylem, whereas this part is called as metaxylem. Both are primary xylem only. The first formed primary xylem is called as protoxylem. Later formed primary xylem is called as metaxylem. So primary xylem is exterior towards periphery, metaxylem is towards interior. Such condition is called as exarch. This is found in roots. I drew the diagram here, dicot root generally, and we find endarch in stems. Endarch means protoxylem is inwards, metaxylem is outwards. That is centrifugal differentiation. This is centripetal differentiation. So let us see what are the conjunctive tissues. Conjunctive tissues are those which are found in between xylem and phloem. They are found in between xylem and phloem. See, I am darkening here circles. These are called as conjunctive tissue. So conjunctive tissue is a parenchyma tissue that is found in between xylem and phloem of dicot root and monocot root. What is their main function? They store food material, and in dicot root, conjunctive tissue as well as pericycle both will form the secondary vascular cambium. Let us suppose this is pericycle. We know pericycle is 2 to 3 layer, thick walled, parenchymatous, living. So this pericycle which is enclosing the stele, when the primary growth is over, before secondary growth, this pericycle will unite with what do you call the conjunctive tissue like this. See this is how the conjunctive tissue and pericycle both will unite and they form a ring of cambium that is called as vascular cambium. This vascular cambium completely secondary in origin that gives rise to outer secondary phloem, inner secondary xylem. Right? In dicot stem, the vascular cambium is partly primary, partly secondary. In dicot root, the vascular cambium is completely secondary. It is formed from pericycle and the conjunctive tissue. You see, when it is formed, it is wavy. It is star-shaped. It is formed from pericycle and conjunctive tissue. It is wavy and star-shaped. And gradually, over a period of time, it will become circle. So this is a peculiar phenomena seen in dicot root during the secondary growth. So remember, conjunctive tissue lies in between xylem and phloem. Lies in between xylem and phloem of roots, not of shoots, found in roots. So that is conjunctive tissue. A particular element, a particular type of cell is taken here in the exam. Third one is interfascicular cambium. What is interfascicular cambium? Remember, in dicot stem, if you see, the vascular bundles are present like a circle. This is called as ring-like vascular bundles. 15 to 20 vascular bundles are present in a circle. It is also called as U-stele. In every vascular bundle, conjoint, collateral, open, that is outer we find the phloem, inner we find the xylem. See, this is phloem outside and this is xylem, that is inside. And in between phloem and xylem, cambium is present. This cambium is called as intrafascicular cambium. Intrafascicular cambium. So intrafascicular cambium lies in between xylem and phloem of dicot stem. So since cambium is present in between xylem and phloem, such vascular bundle is called as open vascular bundle. It is called open vascular bundle. It is found in dicot stem. It is found in dicot stem. Let us see what is interfascicular cambium. See, in between vascular bundles, there will be medullary rays. These medullary rays undergo dedifferentiation. De-differentiation means conversion of permanent cells into meristematic cells is called as de-differentiation. So, due to de-differentiation, the medullary rays undergo, they form into meristem, and this meristem is called as interfascicular cambium. Interfascicular cambium. Right. Interfascicular cambium is completely secondary in origin because it is formed by de-differentiation of the medullary rays, whereas intrafascicular cambium is primary in origin because it is present in plant since its birth. So what happens here? The interfascicular cambium and intrafascicular cambium both will unite and they form a ring of cambium called as vascular cambium. VC, vascular cambium. This vascular cambium will act like secondary vascular tissue in dicot stem, and outside gives rise to secondary phloem, inside gives rise to secondary xylem. But it is more active towards inside, forms more secondary xylem and less secondary phloem. So that is interfascicular cambium. Next, spongy parenchyma. Where do we find this spongy parenchyma? Spongy parenchyma is found in dicot leaf. You know, this is epidermis, upper epidermis, adaxial surface. This is lower epidermis, abaxial surface. Of course, it will have stomata also, even here also present here. Now, the tissue present in between upper epidermis and lower epidermis is called as the mesophyll. In this mesophyll, two types of tissue are present. Upper palisade tissue, which are column-like cells are found. They are generally 2 in rows, and lower we find the spongy tissue. These are loosely arranged mass of parenchyma leaving larger intercellular spaces. So this chlorenchyma, which are loosely arranged leaving larger intercellular spaces, are called as the spongy parenchyma. So the main difference is that this is upper palisade and lower spongy. Both are chlorenchyma only, both contain chloroplast, but number of chloroplasts are more in palisade, less in spongy. And the gaps, intercellular spaces are less in palisade, more in spongy. So upper palisade is meant for more photosynthesis, less gaseous exchange. Lower spongy tissue is more gaseous exchange, less photosynthesis. So because of these two, a complete differentiation physiological anatomically, it is called as dicot leaf, also called as dorsiventral leaf. Right? So like this, in this question, different kinds of tissues or cells are asked here. Subsidiary cell is asked, conjunctive tissue asked, interfascicular cambium is asked, spongy parenchyma are asked. These four kinds of tissues are completely different. They are nowhere connected to each other and all are put together and being they are described on left side, and he is asking which of these four is correct and which are wrong. So you should have a complete understanding of the tissues and their structures as well as functions. Then only you can keep the right answer without any difficulty. First one you see here, large colorless cells in the epidermis of grass leaves. No, they are not subsidiary cells. Large colorless empty in the epidermal cells are called as bulliform cells, or they are called as motor cells. Bulliform cells, or they are called as motor cells. These motor cells are meant for, they are present only in upper epidermis of some monocot leaves, not all. They are all colorless, large, empty, and what is the main function is that when there is no water in plant, they roll the leaf. See the magic. When there is no water in plant, they roll the leaf. When they roll the leaf, stomata get inside. When stomata get inside, then transpiration is stopped. When water is there in plant Again they unroll the leaf. So when they roll the leaf, stomata closed, stomata get inside, transpiration is stopped. When they unroll the leaf, stomata become exposed and transpiration will go on. So it is a defending process. It is what you call a safe measure in some grasses because to protect the plant from water stress. Remember, they are present only in upper epidermis of some monocots, some grasses, not in all. They are called Bulliform or motor cells, but they are not subsidiary cells. So this is not the correct pair. Next you see, in dicot leaves, vascular bundles are surrounded by tissue by large thick-walled cells. See, dicot leaves, vascular bundles surrounded by tissue large thick, they are called as bundle sheath cells. They are called as bundle sheath cells because bundle sheath cells are found in dicot leaf vascular bundle. They have some features that they extend, bundle sheath extensions. Bundle sheath extensions are meant for conducting food material from source to sink, that is from mesophyll to the vascular bundles. Next, so this is also wrong. Second is also wrong. It's not conjunctive tissue. Cells of medullary rays that form part of cambial ring, that is interfascicular cambium. This is exactly right. Third option is right because I have explained you interfascicular cambium is formed from medullary rays by a process called as dedifferentiation. I am writing here dedifferentiation. So by dedifferentiation, the medullary rays, the parenchymatous medullary rays will convert into intrafascicular cambium. Intrafascicular cambium will again unite with intrafascicular cambium. Both will form a ring of cambium called vascular cambium. That vascular cambium outside gives rise to secondary phloem, inside gives rise to secondary xylem. So this statement is correct. Fourth one, let us see. Loose parenchymatous cells rupturing the epidermis and forming lens-shaped opening in bark. Yeah, this is called as lenticels. Beautiful question this is. They are called as here lenticels. What are lenticels? These are lens-shaped openings present in bark. Right. How they are physiologically important? Lenticels will allow gaseous exchange. Behind the bark in dicot stem, we find the cork cambium. Behind the cork cambium, we find the secondary cortex. Secondary cortex is having chloroplast. It will perform photosynthesis. For photosynthesis, it is required to take carbon dioxide and it has to liberate out the oxygen. But the cork cells having suberant cell walls, they do not allow any gaseous exchange. Then what happens? Within the cork cells, some of the cells will rupture due to the pressure and length-shaped openings are formed. It is bark, it's not better it called as cork because bark is different and cork is different. Cork is outer phylum, bark includes even secondary phloem also. Bark includes the cork, carcamium, secondary cortex and even secondary phloem also it includes. So better we call it as cork. So lenticels are meant for gases exchange and they provide the gases required for photosynthesis to these chlorenchyma present in secondary cortex of the dicot stem. So very beautiful question here. Here is asking so many issues, so many concepts are being asked here. If you see very carefully, other than option 3, option 3 is correct because left right both are matching in option 3. But option 1, option 2, option 4, left right both are not matching. Both are different here. Option 1, option 2, option 4. That is 3 options are not matching. So he is discussing here, he is testing your knowledge of 6 options. You should know what are large colorless empty cells in epidermis of gracillus, that are bulliform cells. You should know at the same time what are subsidiary cells. And you should know the dicot leaf vascular bundles enclosed surrounded by bundle sheath cells. You should know at the same time conjunctive tissues. And fourth option you see, Loose parenchymatous cells rupturing the epidermis, forming lens-shaped opening in bark, lenticels you should know. Same time you should know what is spongy parenchyma. So total he is testing here in option 1, 2, 4, 6 concepts he is testing. Have you understand? 6 concepts he is testing, and third option is testing 1 concept. Total he is testing here 7 concepts. You believe it or not, it's a bitter fact. When you are thorough with 7 concepts, you are rewarded with 4 marks. Understand? When you are thorough with 7 concepts, then only you will get the 4 marks. In any concept, either left or right, if you do not have the correct understanding, the very difficult to take this question rightly. So this question is taken from anatomy, histology, that is internal morphology of the plant. I have been trying to explain you everything, but since the space is very less, I could not explain you thoroughly. But orally, I have explained everything. You better go through NCI textbook, that is histology, anatomy. It's very complicated. You should give minimum 5 to 6 readings along with maintaining your own notes with your own sentence and words. Then only you can do these type of questions. Better have picturesque diagram, picturesque memory is better than the written part of memory. So I ask you to draw the diagrams with the neat labeled of all the diagram, then only you can understand this. Better you can copy these diagrams also. I took the diagrams from various resources. So I hope my explanation is very good. This question I like it very much. This is one of the best questions from NEET 2021. So let's continue. Let's knock the door of NEET 2022 with a lot of courage and confidence. So hope you understand this better. Not to forget to subscribe my channel Vivid Botany. We will see the next question next time in our next video. Thank you.